Hello everyone, this is Payal Vaja. In this video, I am going to discuss manufacturing and evaluation of soft gelatin capsule. Preparation or manufacturing of soft gelatin capsule. Four methods are available. First one is plate process. Second one is rotary dry process. Next one is eco-gel capsule filling machine. And last one is no-tone capsule filling machine. First one is plate process. In plate process, it is a semi-automatic method in which a warm sheet of soft gelatin is laid over a plate having a dye pockets. Vacuum is applied from underside of this plate to pull the gelatin sheet into the pocket. The liquid medicament is evenly poured into this dice and another sheet of soft gelatin of similar or different color is laid over it. Finally, a die press is laid over the assembly of these sheets which upon application of pressure seals and cuts up the capsule simultaneously. The capsules are removed and washed with a volatile solvent, and washing is done. Why washing is required? Because washing is done to remove the adhering oil from the capsule. That is the plate process. Next one is rotary dye process. In rotary dye process, it is work on the principle that is developed by Robert Erie in 1933. The capsules prepared by this process have uniformly filled medicaments with accurate doses. That is the figure of rotary dye process. It is a fully automatic continuous process in which the liquid gelatin is made to flow from an overhead tank to form two gelatin sheets on two different rotating drums. These gelatin sheets are conveyed between two rotating dies. Rotating dies are over here from the opposite direction. As the sheets come in between the dies, the metered filling material is injected, which creates pockets in the sheets. Simultaneously, these formed pockets are sealed by pressure and heat and are cut off from the sheets as a capsule. The capsules formed are adhered with mineral lubricants applied on gelatin sheets for easy inner movement. These lubricants can be removed by passing the capsules through naphtha bath, and the capsules are then passed through air dryer under pressure to rinse off the excess moisture. Finally, they are proceed for packaging. Advantages of rotary dye processes. Heavy materials like paste and ointments are easily encapsulated. Next one is solid medicaments are easily processed with suitable vehicles and encapsulated. Capsules are filled with accurately measured medicaments and tightly sealed. Last advantages of rotary dye processes. Dye-colored capsules are formed when different colored gelatin sheets are used. Next process is eco-gel capsule filling machine, or it is also called as stern machine, STERN, stern machine. It works on the principle that is developed by Lecture. It is a unique machine in which powdered dry solid, liquids or a combination of liquid and powdered dry solids can be accurately filled into the soft gelatin capsules. The machine, which consists of a system of rotating dies, on attaching certain small machine parts, it can encapsulate slugs or compressed capsules also. Thus, the soft gelatin capsules so prepared may be of various colors, shape and sizes. Last process of manufacturing of soft gelatin capsule is no-tone capsule filling machine. It is a fully automatic machine in which two gelatin sheets are fed into a pair of vertical dies and rows of pockets are continuously formed over these sheets just when the dice closes and opens. These pockets are filled with medicaments, sealed and then cut off as a capsule from the sheet. The prepared capsules fall in a solvent bath kept at a low temperature for removal of adhering matter. That are all regarding manufacturing methods of soft gelatin capsule. Next one is capsule manufacturing. First step is encapsulation. After encapsulation, tumble drying. In tumble drying, dry, sterile air is forced across the tumbler and remove the moisture from the outer surface of the capsule. After that, supplemental drying. In supplemental drying, after the tumble dryers, the soft capsules are placed on a special tray for a final drying in the drying room for a period up to 48 hours. Next one is sizing. In sizing, automatic capsule sizing machine eliminates undersize and oversize capsule. Next step is inspection. In inspection, it includes visual inspection to check malformed, damaged or improperly filled capsule. It is removed out. Last one is packaging. In this step, capsule may be packed in a glass or a plastic containers or may be strip packed so long as such packaging involves tight closures and plastics having a low moisture vapor transfer rate. That is regarding capsule manufacturing steps. First step is encapsulation. Next one is tumble drying. After tumble drying, supplemental drying is performed. Next one is sizing, inspection and finally packaging of capsule is occur. Quality control test or evaluation parameters of soft gelatin capsule. First one is stability test. In stability test, stability of pharmaceutical dosage form is defined as the ability of dosage form to carry on its physical, chemical, microbiological, therapeutic and toxicological identifications right from the date of formulation and packaging until its therapeutic action is not reduced than a fixed proportion of labeled potency. Stability tests for capsule are performed. Why? Because to know the integrity of gelatin capsule shell, but not to know the stability of therapeutic API or active agent, and for determining the shelf life of capsule, stability test is required. So this type of test help in improving the quality of content of capsule shell and for choosing the appropriate retail package. Before actually performing stability test, the following facts have to be remembered. Which type of facts? The capsule shell are to be sterilized to non-atmospheric conditions with a relative humidity about 20 to 30% and temperature about 21 to 24%. Second condition is the capsules are to be proceed for further test only after they re-establish the equilibrium at room temperature. Stability tests include two types. Which one? First one is self-integrity test and second one is determination of shelf-life. In self-integrity test, this test is performed to find out the integrity of capsule cell. So method of shell integrity test is the capsules which are to be tested and the standard capsules are placed in one of the above condition. Which condition? Like room temperature is maintained and type of container used is open container or 40 degrees centigrade temperature is maintained in type of container is tightly closed container used. For 2 weeks with a periodic examination. The gross and the net charges occurring in them like the standard capsule shell kept at the room temperature and 80% relative humidity becomes more soft, sticky and swollen. The test capsules kept under the same condition undergo the changes. Which type of changes? Gross changes include color fading, any discoloration, disintegration, leaking, or that is turning into brittle or soft. Second change like net changes include loss of volatile ingredients, growing darker, grooves growing darker and wider, and little change in color option. Many times, due to slower drying, soft spots appear over the test capsule which touch the container or other capsule. This type of spots are not developed in a standard capsule. Later on these spots turn harder and are not considered as a defect of capsule cell. In case they do not turn harder due to capsule ingredients, physical stability problem may arise. So, such flaws must be corrected as soon as possible by altering the ingredient of shell or fill the correct material. That is regarding shell integrity test. That is the subtype of stability test. Second subtype of stability test is determination of shelf life. Shelf life or the expiry date of packed capsule is determined under a normal storage condition. Next test is invariability test. In invariability test, weight variation test and content uniformity test is performed. In weight variation test, method is individually weigh a 10 filled capsule. Second step is empty the content of capsules by washing with suitable solvents. Next step, allow the solvents to evaporate from the contents at a room temperature for about half an hour and take the necessary steps to prevent the uptake or loss of moisture. In next step, net content are calculated by weighing the empty capsule cell separately. And final step of weight variation is finally the content of therapeutically active ingredient is determined from the outcome of assay directed in the individual monographs in a pharmacopoeia. That is regarding invariability test. And weight variation is a type of invariability test. Second type of invariability test is content uniformity. How performed? Invariable distribution of drug substance persists as a problem in case of solid dosage form. This problem remains at a higher level in the case of potent drug. For this reason, 30 capsules, according to stated in a monograph, are used for assaying the drug content. So, method for drug content performances or drug content uniformity test is at first 10 capsules are assayed individually to determine the percentage purity of active ingredients. 9 out of these 10 capsules should be within the range of 85 to 115%. In case 1 to 3 capsules are outside the standard range. Standard range is 75 to 125%. Then additional tests are performed on the remaining 20 capsules and the net result of the 30 capsules assayed should prove that at least 27 capsules are within the desired extreme. Example, between 85 to 100 and 150% and no capsule is beyond the standard range. Last test, disintegration test and dissolution test. We already discussed in evaluation test of tablets. Last test is moisture permeation test. In this test, why it is performed? To assure the suitability of containers for packaging capsules, USP, United States Pharmacopoeia, has stated some rules and regulations. According to these rules and regulations, the moisture permeating features of capsules packed in a single unit containers like blister pack or strip pack and unit dose container like glass container or plastic bottles are to be determined. Procedure of moisture permeation test is how it is conducted or performed. For performing this test, one capsule in the single unit container or the unit dose container is packed along with the dehydrated pellets, which have the properties of changing color in presence of moisture. The packed capsule is then placed for a certain period of time in an atmosphere of non-humidity. Any change in the color of dehydrated pellets reveals the absorption of moisture, and the weight of this test capsule is then compared with the weight of capsules under the test. The difference in weight gives the amount of moisture absorbed, that is called as moisture permeation test. Thank you.